It began with a curious pattern in the DNA of bacteria, a strange repetition, a biological puzzle. Scientists didn't know what it meant, just that it looked intentional, structured, almost like a code hidden within the code. Decades later, that mystery would unravel into something far bigger: CRISPR, a discovery that would not only change genetics, but force humanity to confront a terrifying question. What happens when we gain the power to rewrite life itself? CRISPR, short for clustered regularly interspaced, short palindromic repeats, isn't some man-made technology. It's a natural defense system found in bacteria, used to recognize and destroy viruses. But scientists realized they could hijack this mechanism. Instead of letting bacteria defend themselves from invaders, we could use it to edit DNA with unprecedented precision. For the first time in history, we could cut and paste the genetic code like lines of software. This isn't science fiction. It's happening now. We've already used CRISPR to cure genetic diseases in lab animals. We've altered the DNA of embryos. We've grown genetically modified crops resistant to drought and disease. In 2020, scientists used CRISPR to treat patients with sickle cell anemia, with success. The door is open. The question is, how far do we walk through it? Because this isn't just about fixing what's broken. It's about upgrading, designing, choosing. We're not just decoding the instructions for life, we're editing them. And with that power comes an entirely new realm of possibility and danger. Imagine a world where we can eliminate inherited diseases entirely. No more cystic fibrosis, no more Huntington's, no more genetic blindness. But also no more randomness, no more surprises. A world where we design our children for height, intelligence, athleticism. A world where genes are no longer destiny, they're a decision. And here's the catch. Once this becomes available, it won't just be about what's possible. It will be about who can afford it. The divide between rich and poor could become a divide between genetically enhanced and genetically natural. A new kind of inequality, biological, irreversible, and global. We've never had this kind of power before. The power to sculpt life at the most fundamental level. The power to prevent suffering or to reshape humanity in our own image. And the question hanging over it all is ancient. When does creation become manipulation? When does medicine become eugenics? And are we wise enough to know the difference? Because with CRISPR, we are no longer just observers of evolution. We are its engineers, and the blueprint we write might determine the future of our species. To understand the full weight of what CRISPR allows us to do, we need to step back and consider what DNA really is. At its core, DNA is a digital code, 4 letters, A, T, C, and G, arranged in sequences that determine everything from the color of your eyes to the shape of your brain. It's a language, a programming language for life. And for most of human history, we had no idea how to read it, let alone write in it. That changed in 2003, when the Human Genome Project completed the first full map of our DNA. For the first time, we had a blueprint of human biology. But mapping is not the same as mastering. CRISPR changed that. With CRISPR, we can now go into this code, search for a specific gene, and cut it out, like a typo in a sentence. We can delete it, replace it, modify it. But what does that mean in practice? It means that diseases once considered incurable might now be reversible. In 2017, scientists used CRISPR to remove a heart disease gene from a human embryo. In 2018, researchers edited genes inside living humans to treat hereditary blindness. In agriculture, CRISPR has created crops that resist pests without pesticides, that thrive in hostile climates. In labs around the world, scientists are creating pigs resistant to viruses, mosquitoes that can't spread malaria, and yeast that can produce synthetic drugs. The potential is staggering, but so is the risk. Because editing DNA isn't like fixing a broken machine. Genes interact, systems are interconnected. A change in one place can ripple through others in unpredictable ways. A fix might cause unintended mutations. A therapy could become a weapon. And then there's the ethics. In 2018, a Chinese scientist named He Jiankui shocked the world by announcing that he had edited the genomes of twin girls to make them resistant to HIV. He bypassed global consensus. He acted in secrecy, and he crossed a line many believed should not be crossed: germline editing. Changes that aren't just for one person, but for their children and their children's children. Edits that become permanent, generational, irreversible. The reaction was swift and severe. He was sentenced to prison, condemned by the scientific community. But the message was clear: the technology is here. The question is not whether we can use it. It's who will use it first and how far they're willing to go. CRISPR's simplicity is part of its danger. Unlike previous genetic tools, which required specialized conditions and expensive equipment, CRISPR can be used in basic labs with relative ease. The barrier to entry is low. The power is enormous. This means regulation is more critical than ever, but also harder than ever. How do you police a technology that can be practiced in private? How do you enforce ethical boundaries in a global race where not everyone agrees on what those boundaries should be? This is no longer a distant dilemma. The choices we make now about what should be edited, what should be left alone, and who gets to decide will shape the human future. And we are running out of time to make those choices. So what does the future look like in a world where CRISPR is fully unleashed? Let's begin with medicine. In the next few decades, we could see a dramatic reduction in genetic diseases. Conditions like sickle cell anemia, cystic fibrosis, Huntington's disease, each caused by a single gene mutation, could become things of the past. Treatments could evolve from managing symptoms to eliminating causes. Personalized medicine could take on a whole new meaning. Imagine receiving a tailored gene-editing therapy based on your unique DNA profile, correcting predispositions before they ever manifest as illness. Cancer therapies might also become far more precise. Instead of using toxic drugs to blast everything in sight, CRISPR could enable immune cells to be genetically reprogrammed to target and destroy tumors with surgical precision. Already, early clinical trials are showing promise. But it goes further. Gene editing could influence aging itself. Several genes have been linked to longevity, cellular repair, and resistance to diseases. By tweaking these genes, scientists hope to slow aging, extend health span, and perhaps even reverse aspects of biological decay. If this becomes reality, we won't just live longer, we'll live better, staying healthy and active far into what we now consider old age. And what about enhancement? This is where things get murky. CRISPR doesn't only allow us to fix what's broken, it also opens the door to improve what already works. Stronger muscles, sharper memory, higher intelligence, greater resistance to stress or pain. Even aesthetic traits, height, eye color, hair texture could become selectable. Once that door opens, it's hard to close. In a competitive world, the pressure to enhance could become overwhelming. Parents might feel compelled to edit embryos, not just to avoid disease, but to give their children an edge. Employers might prefer enhanced individuals. Insurance companies could charge more for the naturally born. A genetic divide could emerge between the edited and the unedited, the privileged and the natural. It's not science fiction. It's a potential reality. And the consequences are chilling. Because if we're not careful, CRISPR could deepen inequality, entrench discrimination, and even redefine what it means to be human. Some ethicists warn of a future in which genetic perfection becomes a social norm, and those who are unedited are seen as flawed or inferior. We've seen echoes of this before, in eugenics programs, in forced sterilizations, in racial purity ideologies. The danger isn't just in the technology, but in how we use it and who gets to decide. And then there's the psychological dimension. If we can edit everything about ourselves, our looks, our personality, our predispositions, what happens to identity? To authenticity? If a person is designed to be a certain way, do they still choose who they are? These are questions CRISPR raises, not just about biology, but about philosophy, ethics, and the soul. Because in rewriting the code of life, we may also be rewriting the boundaries of human nature. We are standing at the threshold of a new epoch, a time when evolution is no longer driven by random mutation and natural selection, but by intention, precision, and code. The question is, are we ready to cross that threshold? The line between science and mythology has always been thinner than we think. For millennia, humans have dreamed of reshaping life, of healing the sick with a touch, of crafting perfect children, of becoming immortal. With CRISPR, those dreams no longer belong to fantasy. They're experiments in progress, real, tangible, dangerous. But here's the paradox. The more power we gain over life, the more we must confront what it means to use it responsibly. Gene editing is not inherently good or evil. It's a tool, a scalpel that can heal or harm depending on whose hand holds it. So who decides? Scientists, governments, corporations, religious leaders, you? The ethical questions can't be answered in a lab. They require global conversations, inclusive debate, and above all, humility. Because history has shown us that every revolutionary technology brings unintended consequences. Nuclear energy gave us electricity and bombs. The internet gave us connection and surveillance. CRISPR offers healing and the risk of hubris. And yet we can't turn back. The genie is out of the bottle. CRISPR is already transforming medicine, agriculture, and research. It's helping save endangered species, fight pandemics, and grow crops with fewer resources. The potential is enormous, but the price of misuse could be catastrophic. We are no longer passive participants in the story of evolution. We are becoming authors, co-writers of the next chapter in life's design. And with that comes a burden we've never had before, the power to choose what kind of future we want to build. Do we create a world where gene editing uplifts humanity, or one where it divides it? Do we design for perfection, or preserve the beauty of imperfection? Do we play God or play scientist? Or maybe they're not so different anymore. CRISPR has brought us to a place where science and philosophy collide, where biology meets morality and possibility meets responsibility. The code of life is no longer locked in the vaults of nature. It's editable, understandable, controllable. And that means the next great experiment isn't in a petri dish. It's in us, in how we decide to use this power, in how we balance progress with wisdom, in how we remember that just because we can do something doesn't always mean we should, because the future of life on Earth may no longer be written by evolution alone. It may be written by us, line by line, gene by gene, choice by choice.